I won't need to yell, but if I yell really hard, I won't get it. All right. On this miserable day outside, it's wonderful to see Martin here with his, like he's pretending it's Hawaii outside today. So good positive spirit for the end of the week. Look, in the spirit of colour, it's my pleasure to be introducing this exit seminar by Andy Garcia. And Andy has been in the lab for a few years, and as you'll see, also likes colour. Andy is a avid user of Twitter, and if you have followed him, that you will also see that he, during his PhD, is spending many of his hours at odd times of the day, sending us beautiful pictures of wheat growing in the growth capsules. So he's out there at 2 a.m. or 4 a.m. or whatever, and some really gorgeous shots of plants at different parts of the day. So you get to see a bit of that later. How did Andy actually get to be here? That's an interesting one, because it's really quite hard to get foreign students on scholarships into your lab, right? It's not that easy. You know, obviously there's several of you here, and congratulations for being able to be in the room, but it's bloody hard to get scholarships to come to the ANU from overseas. We were lucky that we got a GIDC scholarship that in this case would allow us to actually recruit an overseas student. And that's quite rare because often they're limited to Australian students. It was really excellent that we had, through the ARC Centre of Excellence in Plant Energy Biology, a GIDC scholarship that we could offer to a foreign student, and then the university would then provide some support as well. So really wonderful to have that. But then the question is, where do you get the student? Somebody who's got the right skill set, interest, and so on. And I was really lucky to have a relationship with Kevin Griffin, who's at the University of Columbia in New York, really good friend of mine. We've done a lot of field work together and published a lot together. And he had in his lab an undergraduate student in doing some work on lichors and gas exchange and a bit of respiration in the light and so on. I thought absolutely perfect, right? If he's coming from Kevin's lab, he'll be well skilled and well knowledgeable about the issues that are at play in regards to plant gas exchange, not on wheat necessarily, but certainly would have the broader understanding of the sort of concepts at play. And luckily we're able to recruit him. I see that on your CV, you'll be working through the Gap during that period when you were at Columbia. But, you know, coming to New York City, can you imagine it? You know, that sort of wonderful environment of Columbia University to where is everybody in Canberra? Must have been the feeling that he had. Family lives in Texas, so perhaps it's a bit more like Texas, I'm not sure. So there's a bit more sort of similarity there. Been really lovely to have you in the lab. You're a really lovely, caring person and a really good, nice sort of contributor to the dynamic of the lab. So I'm going to, you know, obviously miss you when you're finally finished and hand in your PhD. In terms of the actual research itself, it's been about wheat and it's been about respiration. It's been an interesting path. And I think most of the students in my lab get to a point at the very end where they go, oh, yeah, it's all clear now. But the bloody journey there was never linear and it was never straight. And I think that's been the same for Andy. It's been getting to a point of a bit more clarity now, but the journey's sometimes been going in this direction, this one, trying to work out what would work and what wouldn't and so on. And I think that the thesis is going to come together nicely. It's still evolving in terms of some of the concepts and so on. But nevertheless, congratulations for getting to this point. Look, obviously, you've all gone through, PhD students over the last few years, extraordinary challenges. And I think the journey going through COVID, to have 2.5 years of the challenge of sort of, you know, on, off with experiments and not being able to necessarily meet with everyone has been a real challenge. So hats off to you for getting to this point, and hats off to everyone else that's sort of near their coming to the ends of their PhD, because I think it's been extraordinary challenge, completely unprecedented and so on. And I think that's been a challenge for us as a lab, trying to work out keeping the continuity of the projects and so on. So again, congratulations for getting to this point. So you're going to hear all about variation in respiration. It's building on, in part, some of the work that Rashmi had done before she headed off to the UK. So it touches on some of the work that's Australian-based and some of the work at CIMMIT, and it explores some of those issues in more detail here in Canberra. So over to you.
All right, well, thank you everyone for showing up on this rainy Friday afternoon. I'll be talking about wheat growth in a changing world, so the genetic and environmental drivers of variation in leaf respiration. So let's get into it. Um, oh, okay. So the story starts with the demand for crops outpacing agricultural output in recent years. So while we see plateauing yield per hectare across the world, obviously with population growth and with growing affluence, the demand for calories is steadily increasing. So you can see here a range of models looking at about a 30 to 60% increase in food demand by 2050. Slightly higher if we take into account climate change as well, which is an important thing to take into account because, as we all know, the world is warming. You can see here projections by 2050, we could be anywhere from 1.5 to above 3 degrees warmer than the 1900s baseline. And by 2100, really up to 5 degrees warmer in the world. And this matters because particularly with wheat, we can see that there is a 6% decrease in yield for every degree Celsius increase in mean growing season temperature. So, 3 degrees warming is about 20% decrease in yield. 5 degrees warming is pretty much 30% decrease in yield. So pretty worrying facts to take into account. And so we have to ask ourselves, if the business as usual approach isn't really getting us to where we need to be in terms of projected agricultural yield, what are the components of yield that we can look at to maybe optimize some parts of it? So there's this equation here by Steve Long and his group that looks at yield potential being, in optimum conditions, what's the ceiling for yield that we can hope for? Which is a function of solar radiation. Plants need light for photosynthesis, as well as light interception. How much of that light is actually being captured by the leaves and taken to photosynthesis? Conversion to biomass. Now that we have light energy and CO2 in the leaf, how do we actually turn that into mass, right? Big, healthy plants. And then harvest index or partitioning efficiency, how much of that mass is actually going into a usable product, right? We don't really need a plant that's 90% roots if we're not going to eat the roots. So if we look at where these factors stand at the moment, you'll see the 3 E's at the end of the equation, E for efficiency. So the efficiency of interception theoretically has a maximum of about 0.9, so 90% of the light that hits the leaf could be intercepted. And we're doing pretty well on that front. We're at about 80% of that. Harvest index is about 0.6 of the plant can be converted into usable materials, and we're doing pretty well on that as well. But conversion to biomass is really only at about half of its theoretical maximum right now. And so if we look at this equation and have to pick one to really work on, it's clear where the target is, right? So there it is. So just a refresher for everyone in the room that photosynthesis and respiration are, balanced reactions more or less. Photosynthesis takes in sunlight, water, and CO2 into the plant and then turns that into carbohydrates. The plant then takes those carbohydrates and respires them to produce energy, giving off CO2 in the process. But in terms of the biomass accumulation, throughout the right side of this figure, respiration, you can take molecules out from certain parts of the pathway, and those are the carbon skeletons that become building blocks for the plant to get bigger, right? So thinking about photosynthesis and respiration in a warming world, what happens to these two physiological processes as the world gets warmer? So on the left, you see a graph with two plants. The blue line is the cold acclimated plant. The orange dotted line is the hot acclimated plant. What that means is that one plant was grown, let's say, at normal cool temperatures, and the orange line was grown at a warmer temperature. So the peak of the curve here for photosynthesis shifts to the right, which means it's met at a higher temperature for the warm grown plant. They can tolerate these higher temperatures a bit better than the cold acclimated plant before falling off as temperatures get higher. Whereas for respiration on the right-hand side, you can see that the cold acclimated plant, which is now the dotted line at the top, increases faster as measuring temperature increases, and it's also higher throughout the course of that measurement. So a warm acclimated plant will reduce that respiratory cost at higher temperatures throughout the range of those measuring temperatures. And so we can start to ask, we picked this efficiency of conversion to biomass as an important target for increasing yield potential. Conversion efficiency depends on the efficiency of these two processes that we just talked about, photosynthetic gains, as well as respiratory losses by the crop. And so in the introduction chapter of my thesis, which is accepted at New Phytologist now, we laid out how we can break down this efficiency of conversion to biomass into actually more pieces. We have alpha, which is the photosynthetic efficiency, as well as the efficiency of energy production and the efficiency of energy use. So alpha, like I said, encapsulates a lot of the photosynthetic parts of the EC term. The efficiency of production covers ATP production, the different oxidases in plant mitochondrial electron transport chains, as well as the different functions of plant mitochondria. And then the efficiency of energy use is, the plant has made ATP through respiration. Now it has to use it for all of these separate processes. Are some plants more efficient in these specific processes than others? And therefore, are they using less ATP? So a lot of considerations. And another way to see it, I think, is to have in this figure here, on the x-axis, you have environmental variables. So you can think of them as temperature, salinity, water availability, any number of conditions that the plant relies on for happy, healthy growth. And in the middle, where that little black triangle on the x-axis is, would be considered an environmental optimum. This plant is growing happily, stress is minimized. And so the shaded green area at the top of this peak, which is the yield potential, is the largest at this point, right? The yield that we're getting out from here is maximized. However, as we move left or right on this x-axis into higher temperature extremes, drought conditions, saline soils, whatever number of environmental stresses there might be, not only does the yield potential decline, and pretty drastically, but the realized yield, which is the green shaded area, also declines because a higher proportion of the energy that's being converted to biomass is being expended in these stress responses that are listed on the right. So my preferred way of thinking about this is to relate it to a bank account, I guess, where we start on the left-hand side of this diagram. The green shaded part of the circle represents the biomass that's gained from photosynthesis. The red shaded part of the circle represents the energy loss from respiratory processes, G for growth and M for maintenance. So one approach we can take is to go the upwards route and increase photosynthetic gain. What does that get us? It gets us a bigger circle overall, but the proportion of energy converted to biomass compared to energy expended is still the same, 50-50 in this example, right? Another way we could do it is to decrease respiratory losses. And so if we take the bottom pathway, we are now keeping the same size circle, same amount of energy coming in, but we're spending less of it on, quote unquote, wasteful processes. And so more is available for plant growth at the end. And ideally, if you get to the right side of this diagram, if you incorporate both approaches, you get a bigger circle that's spending less proportionally. And so in terms of, money in the bank, it's like, do you want more money in the bank? Do you want to spend less money with the money you have, or do you want more money and spend less? And obviously the combination of the two yields you the biggest bank account, which for us would be the biggest yield potential gain. So, it seems like pretty straightforward to say, well, if we make the red part of the circle smaller, that looks like a winner to us. But it's obviously a bit more complicated than that. For example, I'm showing here 138 wheat genotypes that were grown here at ANU and measured using the Q2 automated oxygen fluorescence system. And you can see there's greater than twofold variation across this 138 genotypes. So these plants grown under similar conditions when measured for their oxygen consumption as a proxy of leaf are dark, respiration in the dark, show a considerable spread just based on what genotype they are. And so when we think about leaf dark respiration, leaf are dark, it's a pretty encompassing trait in the sense that you're not really picking up variation in one of these specific processes, phloem loading or nitrate uptake, for example. You're picking up the plant's overall response to these drivers of energy demand, right? So it's not really able to say at the moment that phloem loading, for example, is the huge driver of this demand, but it's able to say we can measure overall that there is higher rates in this one genotype or in this one condition of plants than in another. And so this tool really allows us to compare a large number of different conditions, be they genetic or environmental, in a way that we haven't been able to before. So that sort of brings me to the structure of the thesis. Like I said, it starts with an introduction that's accepted at Neophytologists, where I go over more in depth the processes that make up these photosynthetic and respiratory gains and costs, and also where we are in the field in terms of what improvements can we make to increase that efficiency. We then move on to variation in growth and leaf respiration of 8 specific lines of wheat that were grown in the field in Mexico and here in Australia across different years. That then led me to wonder, what is the impact of growth temperature as well as light conditions on the leaf metabolism? And then finally, I would like to wrap up with a whole plant perspective, right? So a lot of what I talk about today will be covering leaf respiration specifically, but of course, all of the processes going on, nitrate uptake and phloem loading are part of a whole plant perspective, right? There's energy demands all throughout the plant. And so I'll touch on that a little bit as well. So if we start with the wheat that was grown out in the field, this was at 2 sites across 2 different years. One of the sites here in ACT, Ginninderra Experimental Station, out past Belconnen, sort of. And one of the sites out in Obregon, OBR, that's in CIMMYT in Mexico, so the International Center for Improvement of Maize and Wheat Yield. Yes. And across these 4 conditions, 277 genotypes were grown total. So the top left part of this figure shows the different panels being a collection of genotypes that were selected and grouped for a specific trait. So the ones I work with particularly are the 3 at the end, PST, PST sub, and extra. So PST means PS tails, which are the tail ends of photosynthetic capacity. So you have a spread in that trait of how much carbon are they actually taking in. But the interesting thing about this little summary figure is that whether we're looking at just the genotypes on the top left, just the site on the top right, just the year on the bottom right, or developmental stage on the bottom left, there is variation seen across these 277 lines, in some cases more than others. So that brings up the question, and I should at this point take the time to thank Reshmi, Gaju, and all of the team that went out to Mexico for collecting this data, as well as being part of the larger IWIP teams that were part of this project. So that brings up the question, what drives this variation, right? Is it just the genotype? Is it just the environment? If it is the environment, what is it about the environment? Is it that some are grown in hot places and some are grown in cold places? Is it that some are getting more light than others? Or, more than likely, is it an interactive term that combines some of all of these answers? And so here's just a little, excuse me, a little hypothesis chart of what we might expect if the answer was affirmative for each of these. So if it's just the genotype controlling this trait, what we would see on the left-hand side is that when we move from one environment to another, you wouldn't see a change in the trait, but the differences between the three genotypes here listed would be maintained. So the blue one would always be higher, the red one would always be lowest, and they wouldn't change that value due to the environment. If it was due just to environment in the middle chart, you would see that they all have the same sort of value in environment 1 and the same sort of value in environment 2, but there is a difference between environment 1 and 2. So genetically, there's not really any spread, but a change in environmental conditions led to a change in the trait. And then if it was something interactive, like you see on the right-hand side, what you would expect to see is that, if we ranked blue, yellow, red as high, medium, low, is that maintained when we move to a new environment? And so in this situation, it's not, right? The red has the lowest value for this trait at one environment, whereas it has the highest value at another environment. So it's interactive, and the three different genotypes are responding to their environment in different genotype-specific ways. So those are the hypotheses that we can test. And hopefully this figure helps having a look at that. So I've listed 4 environments from left to right. We have Ginninderra 17 and 18, and then we have Mexico 17 and 18. And it's a bit trickier listing 4 environments as opposed to just moving from point A to point B. But hopefully what you can see at each stop on this line is that a lot of the lines do cross over, right? There's no distinct winner in terms of which one has the highest or lowest leaf respiration values. We can't just say, the blue line that's on top at environment 1, which is Muno number 1, is not maintained at the top throughout the 4 environments. So clearly what this indicates is that there is interactive effects of genotype and environment when we're measuring this leaf R-dark trait. And then lucky for me, actually, we got to see a little bit of growth data from the field as well using LiDAR, which is digital estimates of biomass with a big fancy camera that rolls over the field and takes imaging. And what we see here is, throughout the growth of the plant, you can see where they hit vegetative phase at the beginning, exponential growth towards the middle, and then where they taper off at anthesis. So it was nice to have that data to play with as well, but it's not, twofold variation in growth in the same way that we saw twofold variation in leaf respiration, for example. So there's more to it than that, which led me to then say, what happens if we grow them in the glass house, right? We have them out in the field, obviously very different environments for each of the trials out in the field. Hopefully the glass house can minimize some of that environmental variation. And so that's what I did. I was thinking I would pass a giant wheat plant to everyone, but I won't. But I did bring one of these little tubes just so you could see better how the plants were actually grown. So a seed sits on a little mesh screen in that small tube that's being pushed up. The shoots grow upwards, the roots grow downwards into these hydroponic tanks that supply them with all the nutrients they could ever want. And you end up with these really nice, happy plants with happy root systems. So just pass that around. Had to go out to Bunnings to buy meters and meters and meters of PVC tubes and cut them up, but well worth the result. And so what we found after growing them in the glass house as well is that there's still about a 20% difference in the growth rates of the 8 specific lines that I was showing. Those lines were chosen because they were present at all 4 site year combinations. So they were in Australia for 2 years, they were in Mexico for 2 years. Those are the only 8 lines that were present across all of them. So it allowed for a really good comparison across sites and years. But when we move them into a glass house, we still see, a difference in relative growth rates, 30% difference in final vegetative biomass, and a 20% difference in leaf respiration, similar to what that first field figure was showing. But from the larger field data set of the 277 genotypes, we actually got a bit of insight into the respiratory variance and what it could be attributed to. So from Reshmi's larger results, we saw that about 30% of the variance in this trait was attributable to those 277 genotypes responding differently to the conditions that they're in. But well, that's the point I'm trying to make, is the conditions that they're in account for 60% of the variance in the trait. 30% coming from where do they sit in the plots. So, there was a really intricate statistical design that I won't go into that allowed them to say, when the plants are grown here as opposed to there, this is how respiration changes. But the part that's interesting for my results is that 30% of the variance comes from what they called day effects, right? Which is just, we measured today, bright, sunny, happy day. You can imagine going out into the field on a day like today and trying to measure something, you wouldn't really get the same results. And so what we take that day effect of variance to mean is that yesterday's weather can really have an impact on the leaf respiration that we measure today, which leads me to the next part of the talk, which is the impacts of temperature and light conditions on wheat carbon metabolism. So you can see this very pink little picture of sugar and starch extractions that we were doing in the lab. So all of the green ethanol is holding the sugars that come out of the leaf. The starch forms an insoluble pellet. You separate them out, and that's how you can measure starch, which we'll get into in a little bit. But where does the starch come from? So like I was saying with the weather, we'll take the figure on the left and say that on a bright sunny day, the plant is photosynthesizing happily. It's eating up a lot of CO2 and turning that into carbohydrates, way more than it needs during the day. So it stores some of that as starch. And the further you get into the day, the bigger that starch pool becomes until you hit a peak right at the end of the day, which the plant then will deplete throughout the night. It will break down that starch into sugars and export them for energy demands throughout the plant. Take then a day like today, very cloudy, not much photosynthesis going on, probably, because there's just not enough light for them to do that. So you get a depleted starch accumulation at the end of the night. It's a lot lower than you would get in a sunny day. And so what that means is that it will deplete that starch, but at a slower rate. One, the demand for that energy is not as high, but two, it'll still try to hit the end of the night to 0 starch, right? So it's not gonna eat up all of its starch in the first 6 hours and then be left sort of carbon starved. It's gonna control for that depletion. And so on the right, basically, is just a simple schematic of, as light increases on the x-axis, you get more starch accumulation on the y-axis. So Coincidentally, and it's a bit funny because it's a very similar experimental setup to mine, but this paper came out the month after I finished my experiment. So there was no way I would have known that Mark Stitt and his group were doing this. But this paper also looks at the effects of light on starch accumulation and depletion. And so what you see on the first column on the left is how much starch is accumulated throughout the day, which is the white part of the graph. And then how is it depleted throughout the night, which is that gray shaded part of the graph. And on the three different rows of this figure, you have different light conditions. So you can consider the top row to be, well lit and happy plant. You can consider the middle row to be moderately lit, and then the bottom row to be pretty poorly lit. And what you'll notice is the peak for each of those graphs comes lower and lower as light levels are also decreased throughout the day. And yet they still all track towards zero by the end of the night. So it's not like I was saying that they just run through all of the starch available. They actually pace themselves, I guess. On the right-hand column, what you see is what happens if we actually shorten the day. So 12 hours of full sunlight, great, right? This plant has a bunch of starch to play with and eat throughout the night. But if you cut it by half, you'll see that the starch decreases a little bit. The effect on this paper wasn't really as high as decreasing the light levels, for example. But it presents two different ways of creating a phenotype or a range of starch concentrations that we can use to measure degradation rates. So can we shorten the day length or decrease the amount of light received during the day to get different amounts of starch? This. The other hypothesis that we are testing in this chapter is that plants have a total respiration rate that is composed of a maintenance rate as well as an export rate. So there's a cost just to maintain the components of the leaf throughout the night, ion gradients, protein turnover, things like that. And that's invariable, whether the plant was sitting in the sun or whether it was sitting in the clouds for a day, it still has to use this energy to maintain all of these things. Whereas there's a cost to export sucrose, which is what starch gets broken down into. And if you have more starch in the plant and it's always coming down to zero at the end of the night, necessarily you have to be degrading it faster throughout the night, which would indicate higher costs of carbohydrate export. And so what you see on the graph on the right is presumably there is a flat point, which is the y-intercept, where any plant would have to pay that much ATP for maintenance processes. The dotted red line represents what the costs would be for a plant that had low starch accumulation throughout the day. And then for plants that had higher accumulation throughout the day, we would expect to see higher respiration rates as well, which is the solid black part of that line. So that'll be another one of the hypotheses that we test. And so just going back to those tubes that were running around the room, we grew the plants hydroponically again in 3 different growth temperatures, a control temperature of 20 degrees day, 10 degrees night. And then we add 10 degrees of heat to the day temperature to give us the 30-degree day, 10-degree night, as well as adding 10 degrees of night warming to give us the 20-degree day and 20-degree night. And you'll see the 3 light treatments at the bottom right corner of that figure as well, where the control plants were receiving 750 micromoles of photosynthetically active radiation. Shaded plants, which we accomplished by putting a mesh screen over some of the plants, had that light reduced by about 2/3. And then the short day plants received that same amount of light. We just put them fully into the dark halfway through the day. And so now you're stopping that continued accumulation of starch. And you can see the plants in these fancy APPF growth chambers that Owen was talking about. So we'll start with a few biochemical results of leaf chemistry. So the first thing I'll point out on the left is that the leaf mass per unit area changed based on the growth temperature, and particularly with a hot day, which is the purple bar at the far right of that first figure. A hot day led to thinner leaves in a way that a hot night didn't. If we look at the amount of nitrogen in the plants, it also decreases a little bit as well. This figure on the right is on a mass basis, but because the purple bar has less mass in the leaf to begin with, if we had expressed it on an area basis, it actually would be even lower than that. And you'd see a more dramatic difference between the temperature treatments. So nitrogen is a big component of protein, and rubisco is a big proportion of the protein that's in the leaf. So we also thought, what happens if we look at these two quantities? And so on the left, you'll see that warming, day or night warming, reduces the amount of protein that's in the leaf, and it also reduces the amount of rubisco that's in the leaf, which is important again because rubisco is what you need for photosynthesis, which is what builds you up all this starch. And so here we see the actual starch values throughout the night. So like Owen said, here I was, poor me, during lockdown in the growth chambers, measuring these plants throughout the duration of their night. So we get starch values at 5 intervals for each of these treatments. You'll notice that the purple line is a little bit longer because it's a short day. The nighttime for that one actually started 6 hours earlier than the other lines, the orange and green lines. But what you'll also see is this pink line towards the end, which is an extended night. So concurrently to measuring the 3 light treatments, we also had a treatment of plants that was just not exposed to light at all, right? They had 12 hours of a normal night. They were expecting daylight to come, and it never did. They just stayed in the dark for 24 more hours. And so what you'd see there is the starch rates that are found in those plants. Pretty much all the treatments brought it down to 0. And you'll also notice that the 3 light treatments all came down to 0 as well, for the most part. The only other thing to point out here would be that the temperature, which is the different columns, the control is the left-handmost column, and then day warming and then night warming. Day warming in particular seems to have lower starch concentrations to begin with. And so it had lower starch degradation rates at the end of the night, which is consistent with it having lower protein and also lower rubisco content. It's not as straightforward when we look at the leaf respiration numbers. So presumably, as you're going throughout the night, you're degrading your starch. You have less starch in the leaf. So you'd expect the rates to be going down proportionally, right? Which is a little bit of what you see in the control treatment, again, on the left-hand side, the control temperature, I should say. But when you add heat into the mix, it's not really as clean. I'd like to say I have an explanation for that part. I don't just yet, but it's still an interesting finding, at least to say that the initial hypothesis that we had as far as leaf respiration and carbohydrate export having a strong link doesn't appear to be the case here. And so actually, if we go back to that hypothesis, you could almost say that what we found is the opposite of that for some reason. And again, I wouldn't know why, as plants increase their starch degradation rate, the respiratory cost actually comes down according to the results that I saw. It's a bit counterintuitive, and it will require some thought when I start writing that up more thoroughly, but it's an interesting finding nonetheless, particularly how linear the treatments come together. And they do come together in the same order that you'd expect. The extended night, which is orange here, had almost no starch to begin with, so it sits on the left-hand side of starch degradation rate, nearly 0. Whereas the control treatment, which had the most starch available to it, sits on the very right side of the graph. So it gives me confidence that, we're not just seeing a bunch of noise randomly throwing a line together, but I can't really say what it means just yet. One of the interesting bits that I was just going to touch on briefly is that this is, again, data from the field trials that Rashmi conducted, where you can see the relationship of leaf nitrogen with dark respiration rate. And it's pretty strongly correlated. The blue points represent the plants grown in Mexico, and the red points represent the plants grown in Australia. And I point this out with about 5,000 data points on this, only to say that it wasn't worth showing my results for these because there was no relationship. And it might just be that we had, 6 data points per treatment. It might be a cause of just needing more data to get this relation showing up. But nonetheless, we didn't find a very strong relationship between leaf respiration and nitrogen-based traits, nitrogen, protein, or rubisco. So what did we find from this chapter? We found that light conditions and growth temperature do affect how much starch is accumulated by the end of the day, which therefore affects the starch degradation rate, because in wheat, starch degradation does follow that diurnal control, like the Arabidopsis plants that I showed from that example paper figure earlier. The leaf respiration was more variable, but at the very least, it would seem that it's not a first order relationship between leaf respiration and starch degradation. There's more at play in the values that we got for those numbers. So the next part of the talk would be to focus a bit more on these day and night warming effects, but to bring it in from a whole plant perspective. I'm just going to drink some water, excuse me. And so you can see here in this really nice picture that was taken by putting an iPhone on a microscope, a wheat root that's being measured at the University of Adelaide with an oxygen electrode that fluctuates a bit, measures oxygen concentration at 2 different points, and then calculates how much oxygen was consumed by the root at that point. I put the picture in because we unfortunately didn't get to conclude that work in Adelaide because of all the COVID lockdowns, but it was the beginning of where the thesis went as far as considering what role does root respiration play when we're thinking about whole plant carbon metabolism. And so, we'll bring back this figure of warm acclimated plants where the cool plant has a higher value of respiration and the warm acclimated plant has now downregulated that. But what do we think it would do to growth, right? So we saw that the shape of the photosynthesis curves and the shape of the respiration curves are not one-to-one. Respiration continues to increase as temperature goes up, whereas photosynthesis declines once it passes a bit of a temperature optimum. So do we hypothesize that as temperature goes up, the growth would be impacted? And then more importantly, for this chapter at least, do we see a difference in day versus night warming and their effects on growth, right? Does it matter when the plant actually experiences this heat, or is it just a function of overall temperature that it experienced throughout its growth? And so I did a growth analysis using the same hydroponic setup that was passed around, and the same temperature treatments as well. So the 2 chapters go really well together. And you'll see on the figure on the left-hand side, ignore that citation, that the final biomass for the plants that we measured does differ with growth temperature. So that red bar at the far left is a hot night condition where it was basically 20 degrees day, 20 degrees night, give or take. And it had the lowest biomass at 200 milligrams of dry mass on average, where the hot day had a little bit more, and then the control treatment of 20 degree day, 10 degree night had the highest biomass. So heat imposition clearly decreased plant final biomass for both night and day warming. And it also had the same effect on the leaf mass per unit area, which is similar to what we saw in the previous chapter, right? The control temperature has, excuse me, a higher LMA than either of the warm grown plants. And it's a hot night here that particularly decreases biomass and, excuse me, and LMA, which is an interesting finding just because other papers have seen that hot nights particularly affect wheat yield as well. Just very briefly, I'll touch on, if anyone was at the Slatcher Award ceremony where Professor Belinda Medlin was talking about the importance of biomass allocation to her work, because it was done on leaves, stems, and roots, we could look at the proportion of the plant that went to each of these three organs in the three different growth temperatures. And given the change in biomass, you might expect that they would adjust how much of their carbon is going to shoots or roots. But what we see here, at least for the plants I grew, is that they're pretty similar across the board, right? You have the leaves of the plant composing about 40% in all three treatments. The stems of the plant about 20 to 25, and then the roots of the plant, that blue part at the very top of the bar graph, rounding it off, and it hits 1 because that's 100% of the plant. But when we look at the respiration numbers that we got from this experiment, we see that compared to the control temperature, that green point, for the two genotypes that we grew, when you get a hot day, this violet point here, you see a pretty big reduction in whole plant respiration, actually, on a CO2 basis now. So that's the effect of the warmer day. And you see a similar but smaller effect with warmer nights. So like the hypothesis showed, when you grow plants at warmer temperatures, you would expect them to lower their respiratory rates to acclimate to that temperature. So the x-axis here is mean daily growth temperature. So because we added 10 degrees warming either at the day or the night, the points, the red and purple points, fall pretty much on the same part of the x-axis. So you can see that if it didn't matter what temperature or what time of day the temperature was imposed, these points should fall at the same place. But clearly it does matter whether you're warming the wheat during the day or at night. The interesting thing here, and I'll just reiterate that warmer days saw a bigger reduction on a CO2 basis at the whole plant level, which contradicts what we've seen at the leaf level. So many of you knew Brad Posh, no, he's still alive, who was in our lab recently, and his work found that it's the night temperature that's affecting respiratory acclimation, at least at the leaf level, and at least on an oxygen basis. So what you see in this figure is if we take two shapes of the same color, so we'll start with the triangles, for example, the blue and the red triangle at the 15-degree night temperature had different day temperatures. So the red triangle is 6 degrees warmer in the day, but there's no difference in that respiration rate at 25 degrees. Similarly, the red and blue circles had different day temperatures, but they're pretty close to each other in terms of respiration rate. Whereas as we move higher on the x-axis, so higher night temperature, we see that those respiration rates for all treatments are falling, which pointed pretty strongly to night temperature being the driver of respiratory acclimation in leaves on an oxygen basis. And so this last little chapter was interesting because it showed that the growth parameters in wheat are temperature sensitive. You see those changes in LMA, not so much in the biomass allocation. But the other finding of this chapter was that when we measure, the canopy and then the whole plant, when we take into account shoot and root respiration together, they acclimate to warm days on a CO2 basis in ways that aren't seen at the leaf level on an oxygen basis. And so that sort of contrast between leaf level and shoot level gas exchange highlights the fact that there's a lot that we still need to learn when we start scaling up these relationships from, taking a little cutting of a leaf and extrapolating that to represent a whole plant. There's a lot more that we need to do from that. But so if we move back to our starting point, which was this equation of yield potential, and we decided to choose the efficiency of conversion to biomass, which is highlighted in red. We broke that down and we said it's a function of photosynthetic efficiency, the efficiency of energy production, and the efficiency of energy use. I mean, clearly we can see that when we try to look at specific pieces of each component, for example, phloem loading and carbohydrate export, even when we zoom in on that one piece, it's still a pretty complicated process to explain. And it doesn't really, at least from my work, come together neatly in a one-to-one way like we hypothesized. But the important thing to highlight, I think, from the work that we've done is that there's a lot of room to move forward, particularly with the advances in phenotyping that we see recently. So you would have seen a lot of graphs today showing leaf respiration with other traits. It would be more interesting as well if we took the components of leaf respiration, ATP production and energy use, and also compared them to things like growth, right? What's the growth rate of the plant? Compared them to total yield, which is, how much grain are we getting out in the field? It's not really something that I did here in the glass house, but it's a relationship that could be tested moving forward. The other interesting direction to take would be scaling out of leaf R dark into these larger, what's the shoot level? What's the canopy level? What's the whole plant level looking like? Obviously in the field, it's hard to dig up some roots and put them in a machine to measure their consumption of oxygen, but it's becoming pretty clear that that biomass allocation relationship is an important component of figuring out how are these plants responding to changing climate and how are they adapting their yield accordingly. Some of the pieces that I touched on in the introduction chapter that I don't talk about in the presentation today are things like genetic engineering. So editing regulatory elements. One example would be in ATP production. Plants have an alternative oxidase protein that doesn't actually pump protons very much, and so it doesn't make any extra ATP, but it still consumes that energy that's flowing through the electron transport chain. So regulating that maybe only to when it's needed for stress environments would be a big ATP saver in the long run. Things like trait stacking, where we've seen in the Green Revolution, focusing on conventional breeding and specific traits. Dwarf plants, for example, has led to an increase in yield, but those increases are pretty much tapped out. And so we need to start considering a more systems approach. And like I said, all of that could be accomplished through high throughput phenotyping. So the Q2 automated system is where a lot of the results that I showed today came from. There's a lot of work being done to match hyperspectral reflectance to these traits, where you can just clip a leaf, flash some light on it, get some numbers, and plug it into a relationship. As well as digital non-destructive phenotyping, things like the LiDAR, things like mounting cameras in the growth rooms and having 3D imaging, and a lot of nice work that's being done at APPF and other places. And so that does lead me to my acknowledgements. So firstly, of course, I'd like to thank all of the lab members, past and present, that have helped me with this experiment. My supervisory panel, Owen, Steve Tyerman in Adelaide, John Evans here at ANU, and then Nick Taylor at UWA. And then, of course, all of the RSB staff that comes together to help us all with our experiments and make our days just a little bit easier. The statistical layout of the plants was conducted by the now defunct, I think, SCU, and particularly Juan Jin was really helpful with that. And then just everyone else throughout RSB, as well as the funders of my project, the Center of Excellence in Plant Energy Biology, and also the GRDC. So thank you everyone for your time today, and I'm happy to hear questions.